Hello everyone, myself Dinesh Vaishnav. So today we are going to see a small topic of plant physiology, in that photosynthesis in higher plants. We'll be seeing a light reaction in that. Okay. Now, in this light reaction, we will be having the light absorption from the sunlight. Okay, water splitting would be happening and oxygen release. So small reaction you might be knowing from so many years. You have seen this reaction. That's a photosynthesis reaction. 6CO2 plus 12H2O in presence of what? Sunlight and chlorophyll, giving C6H12O6 plus 6O2. Okay. Now, carbon dioxide is responsible for producing what? Glucose molecule. And the splitting of water is happening and producing oxygen. So oxygen released by the plants is given by the water. Okay. Now, photosystem. Here, photosystem we will be having two things, that is PS2 and PS1. Okay. So PS2 and PS1, the names are given on the basis of the discovery, and that's why we are calling it as a misnomer. Misnomer means what? We are not following the sequence of that. We are following the sequence from PS2 to PS1, but the discovery of PS1 might have happened earlier, and that's why we have given the name as photosystem 1 to it. Both the system would be having a common thing in it. We will be having a reaction center. What's a reaction center? It's a chlorophyll A in the center over here. Okay, this is actually forming the whole reaction center, where the excited electrons have been given to primary acceptors. Now, this electron acceptor would be having at the periphery some accessory pigments. These pigments are xanthophylls, carotenoids, chlorophyll B. Okay. So these other pigments other than chlorophyll A are present in the periphery, and what they are doing, they are actually broadening the range. Which range? That's the spectral range. We will be having from 400 to 700 the spectral visible range and absorption range also. Okay, this is a, Vibgyor. This is the 7 color of the rainbow, you also know. Okay, so anything below this violet, we call it as ultraviolet rays. Anything above red, it would be infrared. So this is your absorption, and absorption in the plant purpose I am telling you, we call it as a visible spectrum, 400 to 700 nanometer. In this only the absorption light is there. For PS2, we will be having P680, that is the maximum wavelength where the absorption light is being taking part, and this excitation of electrons are happening. And in PS1, we will be having P700. So wavelength at P700. At this wavelength only PS1 would be happening. But this is the reaction center, accessory pigments in the peripheral region. You can see this one chlorophyll A in the part. So this is reaction center, where you will be having chlorophyll A and rest of the accessory pigments forming antenna complex and broadening the range of the absorption center. Okay. Now this is a pair of chlorophyll A in the center. Light is coming and absorbing, giving to primary acceptor. Primary acceptor for both PS2 and PS1 are different. Okay. We need to remember that part also. Now this is the thylakoid membrane. Thylakoid membrane in the chloroplast, they would be forming like piles one on the another. You can see in the middle, this is oppressed part. Oppressed part, you can see this is a green part, is oppressed part. Oppressed meaning is non-exposed. It's between two thylakoid membranes. Now this oppressed part, it would be having both the reaction centers like PS2 and PS1. Okay, PS2 and PS1. This white part over here, which is a non-exposed part, is exposed part and non-oppressed part. So here you will be having non-oppressed part. Here only we will be having PS1. Now there are few things we need to understand. Here green part is there. Here you will be having PS2 and PS1. We call this as non-cyclic reaction. This is your non-cyclic reaction. PS1, it would be having cyclic reaction. cyclic reaction. We'll be seeing this in detail now, what it is. Okay. Now, Z scheme was given by Hill and Bendel, and where the location membrane of the thylakoid. Okay, presence of NADP reductase enzyme. Now, when we are talking about presence of NADP reduction enzyme in Z scheme, that means in cyclic reaction we won't be having this enzyme. Okay, fine. This is a Z scheme where the sunlight emitted over here, light harvesting complex, it would be having chlorophyll A in the center. Now, splitting of water would be seen over here. Now, this is a non-balanced reaction where half O2 you can see. If we need to balance this reaction, we need to multiply by 2 by both the sides. Okay, H2O gives 2H plus half O2, 2 electrons. Now, here excitation of electrons is happening. This is being accepted by phaeophytin. If the question is asked to you, which is a primary acceptor of PS2, P700, here it is your phaeophytin. This is your electron acceptor. Okay, primary acceptor electron. So here this would be giving it to proton acceptor. Plastokinin is a proton acceptor, also known as proton pump. Okay, where this reaction is happening? It is happening in the thylakoid membrane. Okay, it is happening in the thylakoid membrane. So when we are talking about thylakoid membrane, outside it is what? It is a matrix. What matrix it is? Stroma. Inside what it is there? It is having what? Lumen inside. Okay, so this reaction is happening over here. Okay, this reaction happening over here. So splitting of water has happened over here, this part. Okay, we will be seeing in chemo-osmosis hypothesis also the same thing. Here, proton pump would be taking one H plus ions and giving inside the lumen, forming what? Plastoquinol. Okay, giving it to cytochrome B6F. It's an electron transport system over here where the production of ATP is happening. So while giving this energy, you can see that ATP is forming over here. Next, giving it to plastocyanin. So plastocyanin is the factor which is giving its electron to whom? PS1, P700. Okay, so this would be giving it to PS1, P700. So here you can see ferrous sulfate given to ferrodoxin. Ferrodoxin would be having one enzyme known as FNR, that is ferrodoxin NADP reductase. This enzyme is helping to form what? NADPH2. Okay, so this enzyme is helping to form NADPH2. Okay, this is all about your non-cyclic reaction. Now this is your cyclic reaction where this is happening in the stromal lamellae. Now one grana is here, another grana is here. Okay, so this is what? Both grana are attached by stromal lamellae. So here you can see this PS1 cyclic system. On upper part also you can see, but maximum you will be seeing in the stromal lamellae. Here PS1 is present, but what is absent over there? NADP reductase. So if NADP reductase is absent, okay, now see, if the light is coming, this PS1 would be getting excited. So rather than giving it to NADP reductase, it would be going back to form what? ATP molecule. So this is actually your cyclic reaction. Okay, this is your cyclic reaction. So PS1 light is there, giving it to ferrodoxin, then ferrodoxin to cytochrome B6F, forming what? ATP. It cannot form NADP because NADP reductase is not present over here, and then give it to plastocyanin and back to the PS1. So this is your cyclic reaction, and that is your non-cyclic reaction, also known as Z scheme. Okay. This is chemo-osmosis hypothesis. Now the same thing. Okay. This is stroma outside. This is lumen inside. So light would be falling over here in the chlorophyll. This is your thylakoid membrane. Splitting of water is happening. This is a balanced equation over here. 2H2O, O2 plus 2 electrons plus 4H plus ions. Now here what is happening? This excited energy would be given to phaeophytin, that's a primary acceptor. Phaeophytin giving it to what? Plastokinin. Plastokinin is a proton pump, I told in the beginning. So this proton pump would be taking this hydrogen and throwing it inside. Okay, with this part you can see, we'll take two things over here, stroma and lumen. Okay, here you can see H plus ions are increasing. From proton pump, what it is doing, it is taking the hydrogen proton from the stroma and throwing it inside. So here also you can see that H plus ions are increasing. This would be given to cytochrome B6F. This would be passing it to plastocyanin, which is the factor for passing the electrons to PS1 700. So here it is giving into ferredoxin, that is FNR. This is an enzyme which is responsible for forming what? NADPH2 molecule. So this is also a form of energy, you know. Now here also you can see, to form NADPH, FNR is using what? H plus ion. So again what is happening? H plus ions is decreasing in stroma and increasing in lumen. So due to this, what is happening? Stroma would be having the less H plus ions, that the protons are less, and in lumen the protons are more. To balance this proton, we will be having a facellated diffusion. Diffusion means what? Traveling of energy from higher concentration to lower concentration. But we will be having a facellated diffusion, that is your CF0 F1 particle, and see this C stands for chloroplast. Okay, C stands for chloroplast. Okay, F0 F1 particle, again you can see in another chapter also, where you will be having the membrane after the Krebs cycle. You can see that F0 F1 particle where ATP synthesis is there. We are just adding C over here, that is a chloroplast. Okay, now here also ATP synthesis is there. Now what is happening? This everywhere you can see H plus ions is increased. Now this H plus ions would be passing from lumen to stroma. Okay, for balancing from lumen to stroma, it would be going and it would be balancing. While balancing, this H plus ions is having energy. This H plus ions is having energy. So this ATP synthesis, what it will do, while letting pass this hydrogen from inside to outside, it would be taking this H plus ions energy, that is ADP plus IP, forming into what? ATP molecule. So this would be forming ATP molecule. So with this, ATP synthesis is helping for production of ATP molecule, and this gradient is actually helpful for production of energy. F0 particle would be there in the stroma, F1 particle would be there in the stroma, so this would be in this lumen, in the membrane, and this would be in the stroma. This part would be outside, it not be inside. Okay, so this is all about the chemoosmosis hypothesis, and with this we end this part. Thank you, students.